So this is going to be our first time trying out this new flipped class model. I'm very excited about this. The whole point of this is that, I watch this video as our asynchronous assignment. You'll take notes, you'll answer some questions, and then afterwards, once we are in class, we can jump straight into discussion and start practicing with the material and getting more prepared for our exams and for the AP test in May. And so sometimes I talk really fast and I can zip through this material super quickly, and your little arms can't catch up. And so this is a way for you to pause me, mute me, slow me down, speed me up, do whatever you need to do to be able to take notes at your own pace while also staying on track with me. So hopefully this finds you well, and let's get started. So we're gonna be starting our newest unit, Unit 2. The first part of this unit, we're gonna spend talking about cell structure, function, and so basically what are organelles, what do they do, what are their jobs, why are they important, what happens if they're not working. We're gonna be talking about all of that. And so this is gonna be a very vocab heavy section of the class. And I'll explain some resources that we can use later. So before we start talking about cells, we need to start talking about the two major types of cells. And so what I'm going to be doing is that I'm going to be highlighting key vocabulary in pink. And so you should have this vocabulary either in a Quizlet, a note card. You should highlight it in your own notes if you're taking personal notes. Just note these are key vocabulary, whatever I'm highlighting in pink. And so the first two vocabs that we're going to talk about, vocab words that we're going to talk about, are prokaryotes and eukaryotes. These are the two major types of cells that exist. If we were to take the vocabulary and break it down to its roots, pro means before, and karyotic is going to be nucleus. When we look at eukaryotic will mean nucleus, and then U is going to mean true. We're going to be talking about what you're going to need to know is the differences and similarities that exist between prokaryotes and eukaryotes. And we're going to go through them right now. And so right here, these are all going to be the similarities. These are all the similarities. Similarities. I don't promise that I'm going to spell anything right on this. Similarities of prokaryotes and eukaryotes. And so they all have a plasma membrane. All cells have a plasma membrane, a membrane that separates the external, the outside, from the internal, the inside. They're all going to have a cytosol, which is that fluid-like material that's inside the cell. This is where all of the organelles and the nucleus are floating around. It's the fluid that's inside the cell. Think of like a water balloon. If you were to pop a water balloon and water were to gush out, the cytosol is kind of like that water. All cells are going to have chromosomes, which contain the genetic material to make new cells. And they're also going to have ribosomes, which are important for making proteins. These make proteins. The sooner you have this vocab word memorized, the better it's going to help you in this class. Ribosomes are in charge of making proteins. And I'm just going to write genetic info here with chromosomes. This is genetic information. So the four similarities between prokaryotes and eukaryotes is that they have a plasma membrane, they have a cytosol, they have chromosomes, and they have ribosomes. And so it's one thing to be able to list these four differences, these four similarities, but it's another thing to be able to explain it. And so on top of having to know like that they have a plasma membrane, you need to know what a plasma membrane is and what does that mean? What is a cytosol and what does that mean? Let's highlight this one. This is a key vocab word that's going to come up pretty often. Cytosol. And what does it mean to have a ribosome? What do they do? Etc. And so now let's start talking about the differences between prokaryotes versus eukaryotes. Differences. First off, prokaryotes are going to consist of anything on the evolutionary tree that contains bacteria and archaea. Archaea are things that are extremophiles, which means they love extreme environments. They love to live in like methane-filled vents, hydrothermal vents, super cold environments, super hot environments, super toxic environments. They love the very extremes. And this is going to also consist of bacteria. Prokaryotes are going to lack a nucleus. They will have no nucleus. They will have no nucleus, whereas a eukaryote will have a nucleus, does have a nucleus, has a nucleus with a nuclear envelope. So basically a membrane around the nucleus. Prokaryotes are going to have no membrane bound organelles, which means that they just have ribosomes. They only have ribosomes. No mitochondria, no chloroplasts, no endoplasmic reticulum, primarily just ribosomes. There's always going to be exceptions. For example, cyanobacteria, they have chloroplasts in them, but this is going to generally, in the general sense, no organelles other than ribosomes. All the cells are going to be unicellular, which means they are only made up of one cell. They are made up of one cell. And these came before. Prokaryotes are evolutionarily older than eukaryotes. If we were to look at this evolutionary tree right here, we can see that at some point, eukaryotes, this branch right here, eukaryotes branched off from prokaryotes, and archaea branched off from eukarya as well. And so we can see that archaea and bacteria are older. These are older. This is newer. Eukarya are newer evolutionarily. So going back to prokaryotes, their DNA is going to exist in a single strand, or it's going to be in a circular form called a plasmid. Now let's look at eukaryotes. Like we said, eukaryotes do have a nucleus. They're going to be bigger, more complex, and they tend to be multicellular, which means they have multiple cells. They're made up of more than one cell. Like us, like plants, like your dog, like birds, like most living creatures. They're also going to have these membrane-bound organelles like mitochondria, chloroplasts, lysosomes, endoplasmic reticulum, etc. And examples of these are going to include animal cells, plant cells, fungi, slime molds, you name it. And so now we're going to start talking about organelles. Organelles are just tiny little organs that exist inside our cells. This is going to be a lot of vocabulary. I'm just going to be frank with you. This is going to be a lot of vocabulary. Some really good resources is if you still have your cell book, rip out your cell book from freshman year. I bet Miss Martinez would be so proud of you if you still had it. This is going to be a great resource if you have it. What else can you use? Quizlet. Quizlet. I can't even spell Quizlet. Quizlet. I think there's two Z's. What else is there? Cahoots. Flashcards. You need to have these vocabulary memorized. This class assumes you already have this information in your head from biology. I know it's been a couple years since you've taken the class and you probably don't remember most of these, but we need to get these definitions back in your head because it's not just understanding what the cell membrane is or that the mitochondria is the powerhouse of the cell. It's more than that now. You need to be able to understand what happens when the ribosomes don't work. You need to know what happens when how the Golgi apparatus is works in relation to the endoplasmic reticulum. And so you're going to start comparing and contrasting and seeing what happens when something doesn't work. And so it's going beyond just those understanding the definitions of these words. It's going deeper than that now. So we're going to talk about two different classifications of organelles. We're going to talk about the endomembrane organelles, and we're going to talk about energy-making organelles. I'll leave this here. We'll go over each of these individual organelles in just a moment. And so cells are what we call compartmentalized. They go through it's a hard word to say, compartmentalization. And this means that organelles allow for different metabolic reactions that occur in different locations. So I like to compare this to a house, not your house specifically, but to houses. Houses tend to have a kitchen, a living room, bedroom, master bedroom, bathroom. There's all these different rooms in the house that all have different roles and jobs. And it's just like a cell. Within the cell, there's different sections of the cell that have different parts to it. And so, for example, here we got the Golgi apparatus, I believe. We got the endoplasmic reticulum. We have mitochondria. And so we can see that these parts are in certain sections of the cell. Why do we do this? It makes it easier for reactions to happen. It means that organelles are kind of staying in their lane. They're not crossing paths too often and getting in each other's way. By having so many different types of reactions happening in so many different places, we are increasing the surface area for reactions to occur. So that means we can do a lot more things when each organelle is doing its own job. Before we start talking about all the organelles, let's talk about some unique organelles that you're going to find in plants versus animal cells. So with plant cells, let me go back. I did not highlight this key vocab word. I said in pink. Compartmentalization, key vocab word. And so in plants, we're going to have chloroplast, the central vacuole, the cell wall, and the plasmodesmata. We will go over these in either this week's class or next week's class. So don't worry about having these terms memorized right now, like this specific slide. If we mention it this week, then that it's important. If it's not mentioned this week, it will be mentioned next week. And then animal cells, you're going to have lysosomes, centrosomes, and flagella. So now we're going to start talking about the endomembrane organelles. The endomembrane organelles are a group of membranes and organelles that exist within a eukaryotic cell, and they all work together, key vocab word, they all work together to modify, package, and transport lipids and proteins. So their job is to make and send out lipids and proteins. So when we're looking at this, you should be like, okay, this is in charge of making so think of like, what is the most important lipid that exists in a cell? Lipids make up the phospholipid bilayer or the cell membrane. And so these lipids are going to be helpful with making the membrane. And these proteins are going to be helpful with making enzymes for reactions to happen. And so these are just two things that are very important that cells that are created during the endomembrane system or through the endomembrane system. And we're going to see this pathway that's happening here. We're going to start off inside the nucleus, specifically the nucleolus. We'll eventually leave the nucleolus into the nucleus, leave through a nuclear pore. We'll find ourselves hanging out in the endoplasmic reticulum. Eventually afterwards, we will get sent to the Golgi apparatus. Once we're done hanging out here and we're being modified and tagged and shipped out, we will either be sent to the outside environment to do some sort of job or to go to some other cell, or we will be sent to the lysosome or some other part in the cell as well. And so it's a lot of packaging and processing that's happening here. And we're gonna go through this process right now. And so our first key vocab, our first stop is gonna be the nucleus. The nucleus. You should know this one. You should be pretty familiar with this. The nucleus is where chromosomes are gonna be stored. Typically, chromosomes exist uncondensed, kind of like uncooked spaghetti, or like cooked spaghetti, or they could exist like, think of like when you leave your headphones in your pockets for too long and they get all tangled up. They're all messed up like this. DNA just kind of floats around inside the nucleus in here. Around the nucleus, we're going to see this thing called a nuclear envelope. It is the membrane that surrounds the nucleus, separating the inside of the nucleus from the outside. Nuclear envelope. Just give me a second. And then let's write nuclear pore. That's another key vocab. Nuclear pore, nuclear envelope, and nucleus. And so what I'm labeling right now is the nuclear envelope. I'm going to draw the nuclear pores in, let's say, purple. So these little dots that we see all around the nucleus, around the nuclear envelope, these are nuclear pores. And this is what regulates what comes in, who comes in, and who comes out of the cell, or sorry, out of the nucleus. And now you're probably thinking, what's that big red dot that's in the middle of the nucleus? Well, we're about to go over it. That's the nucleolus. Nucleolus. Like nucleus, spell a couple more letters. So the nucleolus is this dense region right here that I'm highlighting in green. And this is where ribosomal rRNA is going to be created. So rRNA is going to be created here. Eventually, this rRNA is going to combine with proteins to form large and small subunits called ribosomes. And again, remember, these ribosomes make proteins. And so these will ultimately be making proteins. They are made from proteins and they will make proteins. And so our RNA is going to come out of the nuclear envelope, out of the nucleolus, enter the nucleus, and leave out the nuclear pore and go into the cytoplasm or to its next destination, wherever it may be. Here. That. So like I said, these ribosomes are going to help with making proteins. So if we can recall back to protein synthesis, this is going to be the site of translation. And this is where the RNA code is going to be read, and we're going to start making those polypeptide chains from the mRNA strand. And so we're going to start seeing like methionine, tryptophan, cytosine, with those sulfur creating bonds. We'll see other molecules, and we'll just keep creating these polypeptide chains that will eventually have like a secondary structure with beta plated sheets and alpha helixes,
Eventually it'll form a tertiary structure and then a large quaternary structure. And so this is where it all begins. We can trace back proteins to the ribosomes that are created from the nucleolus, going back to the nucleus and going back into the nucleolus. It all starts here at the nucleolus. Going back to the nucleus, one last thing. I think it's gonna be really helpful for y'all to figure out roles and titles for each of these different organelles. And so, for example, the nucleus, it to me is kind of like the librarian or like the principal librarian. I should know this, Bolis. I was once a librarian. And so this is where all the information is going to be stored. He is the librarian. He has all the information. He has the books. He has the genetic code, which has the instructions to make every single part of the cell. And so I would start thinking of titles or things to compare these organelles to, because ultimately it's going to help you if you're understanding so much more. It's going to make it so much easier than memorizing random vocab words, especially with the variety of functions that they may have. So ribosomes, we've been talking about them so much. I'm going to highlight them again. I think they are so important. What are they made out of? They're made up of RNA and protein. What do? They make proteins. Their job is to make proteins. That's super important. So we should know what they're made of and what do they do. And where should we find them? And so we're going to find them in two different places. We'll find ribosomes in either the cytosol, which is that liquid-like substance that membranes tend to float in the cytosol. And here, these proteins or these ribosomes are generally used to make things like enzymes. Or we can, if they're not in the cytosol, we're going to find these ribosomes bound to the rough endoplasmic reticulum, the rough ER, or the nuclear envelope. We'll go over what the endoplasmic reticulum is in just a second. And so what's going to happen here is that we'll create proteins at the rough ER, and these proteins can be secreted out of the cell via transport vesicles. Again, don't worry about these vocabs right now. We'll go over these in just a moment. But eventually these will get packaged and sent outside to the external environment. Going back to the endomembrane system, we started in the nucleolus. Right image, where are you? We started in the nucleolus. We leave the nucleus through the nuclear pores, and now we are here in the ER. We are in the rough endoplasmic reticulum, or just the endoplasmic reticulum. As we're going to talk about, there are two different types. The key word, endoplasmic reticulum. What it is a network of membranous sacs and tubes. And the whole point of having this is to compartmentalize the cell, to keep proteins that are found in the rough ER separate from the cytosol. And we're going to have two parts. We're going to have this rough ER, and the smooth ER. And so what the rough ER is going to do is that the rough ER is going to be making proteins. Makes proteins. These proteins can be used for a variety of functions, like making enzymes, antibodies, structural proteins, integral proteins, a lot of things. Whereas the smooth ER going back to the rough ER, if we look at the rough ER, we're going to see these dots surrounding the structure. And what these dots are ribosomes. Think of like little pimples or like little spots on them. This is what gives the ribosome, the endoplasmic reticulum texture. And so it gives it this like rough kind of feeling. Whereas the smooth ER, looks smooth, no texture, there's no ribosomes on it. And this is gonna be the site of lipid synthesis and steroids, which are both lipids overall. And so a lot of these lipids and steroids are gonna be used for making membranes. Super, super important. And so our next structure is gonna be the Golgi apparatus. I don't know why this says complex. No. Apparatus. You're using key vocabulary. I did not spell that right. I am so sorry. Someone call me out on it later. What is it made out of? The Golgi apparatus is a flat membranous sac, a bunch of flat membranous sacs that are called cisternae. And so the cisternae, key vocab word again, cisternae is going to be these individual ones. So we see one let's use a different color. Let's use green. We got one cisternae, two cisternae, three cisternae, 4, 5, 6. This number can vary between cell to cell, but they are the individual sacs. Each of the cisternae are not connected to each other. They are floating independently, and they have a directionality. And so this section right here, the one with the little C, this is gonna be the cis face. The cis face. The cis side. And this is where it's gonna receive messages. This is the front door. And then out here is gonna be the trans face, which is kind of like the back door. This is where it sends messages. So receives, sends. So this is receiving messages from the ER. So when we leave the endoplasmic reticulum, messages will come here into the Golgi apparatus. And then it's gonna be sent to either the cytosol or out to the plasma membrane for secretion. And what I mean by secretion, being sent outside of the cell. Commence secretion. I did not like that. That was weird. Sorry. Yikes. And so what does this do? And so it's going to receive these messages from the ER. And it's going to modify the materials that it gets. And so it's going to be getting ribosomes. And so what it's doing is that it's going to modify and sort these ribosomes and package them so that they are going to the correct place. And they're ready for the job of whatever they're going to be doing. And so I lied, it doesn't have to be just ribosomes, but it could be a lot of other things like lipids and CAHS. And then what it's going to do is that once it receives the message, it sorts it and modifies it. It's going to package it next, and then it's going to put it into a vesicle, which is kind of like think of like a garbage can. A garbage can that gets sent to the outside via exocytosis. And so this is basically excretion outside of the cell. The fancy word for excretion outside of the cell. Think of cell pooping. That's like what I think about it. It's cell pooping. Exocytosis. So there's a couple of final stops for materials. One stop could be the lysosome. The lysosome. The lysosome is a sac that has a bunch of hydrolytic enzymes. And what it does is that it hydrolyzes macromolecules. And so let's think back, what does hydrolyze mean? Hydrolyze means to break down from polymer to monomer. It's breaking down macromolecules in cells. One example is autophagy. This is when it's taking its own, for example, mitochondria, breaking up the mitochondria into its lipids and proteins and carbs, and then recycling its own body parts so that it can use it for new things. For example, making a new mitochondria or making new proteins. Another type of lysosome is called a peroxisome. It's similar to lysosome. It dissolves things, but you're going to see a lot of H2O, a lot of let me double check before I commit to saying this. I want to say hydrogen peroxide. Hydrogen peroxide. Yeah, I was right. Hydrogen peroxide, H2O2. These are very toxic and acidic materials that could dissolve our body parts. And so it's important for this waste to get collected and get put into these peroxisomes, and they don't, so that they're not harming our cells. Think of them as like waste containers. Not even that. Toxic waste containers. And so they're going to turn that hydrogen peroxide into water, something that's safer for the cell to use. Next, we have vacuoles. Vacuoles are going to look different between plants and animal cells. And vacuoles can look like large vesicles that they can be used for transport. They can use for the storage. There's a couple different things. So we'll talk about first the food vacuole. Food vacuole. Let's use a different color because it's getting weird. Let's highlight in green. Food vacuole. And so we're going to see this thing called phagocytosis. This is when the cell is eating something. And so when something gets eaten by the cell, it's going to enter the food vacuole, and that's where it's going to get stored. And then it'll be digested by lysosomes eventually. We'll have a contractile vacuole. It's to maintain water levels. We'll talk about more about this next week. And then most importantly for plants, the central vacuole. The central vacuole is what gives plants their shape. If the central vacuole is empty and there's no water, our plants are going to be sad and limp. Sad flower. Empty vacuole, empty central vacuole. It's empty, there's no water in here, no water. And so when you see your flowers limping like all sad, ooh, aw, it means it's thirsty. And so once you put water, let's say like a little watering bucket, we're gonna have a happy flower that is turgid, meaning full of water. Its central vacuole is full of water, full central vacuole. Super happy flower. Turgid plant cells. Full. So now we're gonna start talking about the energy organelles. And before we start talking about them, we need to start talking about how they first came about. And so we have this theory called the endocybonate theory, or the endocytosis theory. And this, what it says is that the similarities between mitochondria and prokaryotes, how it explains the similarities that mitochondria and chloroplasts have with prokaryotes. And what this theory states is that at some point in early evolutionary time, some hungry eukaryotic cell ate a prokaryotic cell, engulfed. Let's highlight embryonic theory, endocybonic theory. And so what happened is that prokaryotic cell became endocybionic. It started living inside the cells. Think of like symbiosis, when things can live and coexist with each other. And they eventually became a functional organism. Some pieces of evidence that say this is true, some pieces of evidence include that all mitochondria and chloroplasts have a double membrane. They also have their own ribosomes. They have DNA, and they are capable of functioning on their own. A cool side tangent. So we know that our parents give us, at first we start off as one little sperm and one little egg, one giant egg. Mom, dad. And so what your dad brings to the table, he just brings 1/2 his DNA. So does your mom. She brings 1/2 DNA. And then they'll eventually combine to create you. But she also brings the organelles and the cytosol. And so one organelle that she brings is mitochondria. And that is a energy producing organelle. Mitochondria have their own DNA. And so if your mom is giving you her DNA, or she's giving you her, her mitochondria, those mitochondria cells, those mitochondria in your body are 100% your mom's DNA. Although your mom gives you one half of your DNA and your dad gives the other half, this is just your DNA, your nucleus. But when it comes to DNA inside your mitochondria, there are diseases, there are symptoms, there are all these like mutational things that could happen with mitochondrial DNA. It's still being explored to this day, but all 100% of your DNA from your mitochondria comes from your mom, because she gave you all of your organelles when you were first conceived. So the little endocytosis in a nutshell theory. So we had 2 independent bacteria, 1, 2. You could say this one was like had the mitochondria, mitochon. Eventually he was engulfed, he was eaten and got put into his tummy of the big guy. Eventually they were like, whoa, this is working out. Our relationship has gotten so much better. I am my new self. And it ended up figuring out how to make this little guy inside him and be able to synthesize him on his own, and eventually just became a part of the bacteria. And so that's a theory of how eukaryotic cells started becoming a thing. Fun, right? Okay, let's start actually talking about these energy producing cells. Organelles, my bad. And so the first one we're going to talk about is the mitochondria. Not to be mistaken with the mitochondriana. No, mitochondria. Oops. I've literally been waiting this whole lesson just to make that joke. This mitochondria is going to be the site of cellular respiration. This is where ATP is made. ATP production, where we make energy. We're going to have a double membrane inside the mitochondria. The inner membrane is going to have folds called cristae. Cristae. That's not what I want. Do what I want.
There we go. Again, cristae, it's gonna be these guys right here. These are the folds, the cristae. And what this cristae does is that it divides the mitochondria into two internal compartments, and it increases the surface area so that more energy can be produced. More surface area equals more ATP production, which is what we exactly want. The mitochondria also has a mitochondrial matrix. So this fluid that's in the center right here that I'm highlighting in green. This is where we're going to see the Krebs cycle. We'll talk more about this during cellular respiration in the next unit, so don't worry about that for now. This mitochondrial matrix is going to have enzymes, it's going to have ribosomes, and it's going to have our mitochondrial DNA in it. But just know there's two parts. There's the intermembrane, this part right here, and then there's the mitochondrial matrix. The mitochondria, the number of mitochondria in our cell correlates with metabolic activity. And so what I mean by that, cells with higher metabolic activity will have more mitochondria. What is an example of a cell that requires a lot of metabolic activity? We're using it all the time. Our muscle cells have tons of mitochondria. That's not how you spell cells. So the next cell that we're going to talk about is the chloroplast. The chloroplast is going to be the site of photosynthesis. Photosynthesis. The chloroplast is going to have pigments, little green pigments called chlorophyll, and inside the cell, inside of the chloroplast, we'll see some thylakoid sacs with thylakoids of granisacs that will be important for light-dependent reactions. Right now, just know what the chloroplast is. It's important for photosynthesis, and it has pigment. We will go over this stuff right here in a later class, but do know site of photosynthesis has chlorophyll and only in plants. Well, it's also in some like cyanobacteria and other things, but primarily only in plants. Again, we'll talk about this stuff in a later unit. And so let's start talking about the cytoskeleton. We're wrapping this up now. The cytoskeleton is like the skeleton, the bones. The skeleton of the cell. There's going to be 3 layers of this skeleton. Before we start talking about those different layers, overall, these are all similar properties of all three types of cytoskeleton. There are a bunch of fibers that move throughout the cytoplasm, and they give structural support and mechanical support. So if I asked you, what do I mean by structural support? We'll talk about that in a moment. With mechanical support, it gives anchors, and it anchors the organelles, and so that the organelles aren't jumping around inside the cell. So let's say like this is our nucleus. Let's say this is our mitochondria. What the cytoskeleton does is that it prevents the mitochondria from just like floating around, jumping around. playing ping pong or whatever it's called. What's that game? Where you take a machine and you put a quarter in and it like bounces things around. Pinball. Pinball? Yeah, pinball. It's not like a pinball machine. What the cytoskeleton does is, I'll draw this in blue, it keeps it loosely in track. So like I like to think of it like the springs of like a mat, like of a trampoline. You know like how a trampoline is like this, and then there's little springs that go around it. Yeah, you know, like this is a terrible drawing, but let's just let's not go about that anymore. So what the cytoskeleton does is that it anchors in organelles. It allows for the movement of vesicles throughout the body, and it allows for movement to happen with motor proteins. And so this allows the cell to move around actually in our bodies. And so this might include flagella, and we'll talk about the other type in just a moment. So there's three types. We're going to talk about microtubules, microfilaments, and intermediate filaments. We're going to go in order. So microfilaments are, what I mean by order of size. These are going to be the smallest boys, smallest little guys. Microfilaments. The microfilaments are going to be made up of rods of protein actin. Their main, I'm gonna be frank with you. You probably don't need to know what these are made out of. The key understanding of this section is to understand the functions and what makes them different structurally, functionally. And so what they do is that they maintain the cell shape. They keep tension. They help with muscle contraction and cell motility, which means movement. Motility is just a fancy word for movement. And this helps with cleavage during cell division, when our cells finally are trying to finally break apart. That's cleavage. That is what microfilaments help us with. I'll leave this for a second. Next, we're going to talk about intermediate filaments. These are the medium-sized ones. Excuse me. So intermediate filaments are going to be the medium-sized one. Medium. Medium. The medium-sized filaments. These are fibrous proteins made up of varying subunits. They're more permanent structural elements. They're not meant to be broken down frequently. They're there more permanently. Their functions are to also help with cell shape. They anchor the nucleus specifically in other organelles, and they form the nuclear lamina. This is the lining of the nuclear envelope. That's what the lamina is. Not a key vocab word. Not as important as understanding what an intermediate filament is. Next is microtubules. Microtubules. Don't let the word deceive you. Micro, this one is actually the largest one. Still pretty tiny though. This is the largest. This is going to be made up of a hollow-like tube made up of protein or tubulin. They grow from the centrosome, and they help with making microtubules, which is funny. They help with making themselves. They give structural support. Think of like tracks. I like to think of this as the highway of the cell. The highway. Life is a highway. For the move. So this highway acts as allows for the movement of organelles that need to interact with motor proteins, and they help with the separation of chromosomes during cell division. And this is where we're going to see cilia and flagella come into play. So when we talk about flagella, let's think of a sperm. Flagellas are the tails of the sperm. Other bacteria have it, but this is the more classic example. If we're talking about cilia, let's highlight these two, cilia and flagella, key vocab words. The cilia, let's draw a cell. The cilia are going to be these tiny little arms that extend out of the cell, and they allow the cell to slowly move, kind of like a starfish. And so I'll leave this here for a second, sorry. Okay. And so this last slide, this is just giving you the differences between the three different types of cytoskeleton. Sorry, I am thinking a lot faster than what I'm talking a lot faster than I'm thinking. So it's gonna be important for you to be able to differentiate these three and compare and contrast them. And so this is a good slide as a resource. So like I said, there's a ton of vocabulary in here. Cytoskeleton, microtubules, microfilaments, intermediate filaments, chloroplasts, mitochondria, cystae, a lot of things. I really recommend getting note cards, getting a Quizlet going, start helping each other out, practice each other studying, because understanding these vocab words are just the root of this class, and we are going to need to go a little bit deeper than just understanding the general vocabulary. We need to understand it and how to apply it. So I hope this video did you well. At the end of this assignment, you're going to see a question that I need you to ask. Ask a genuine question about this material. What are you confused about? What do you need me to go over? Because this is what we are going to discuss in class tomorrow or Friday, depending on the period. So use these questions as a way of structuring our class. And I will explain this more when I see you again. I hope you all have a good day. Stay safe. Toodles.